Pemirsa, dengan perkembangan bioteknologi yang sangat pesat, khususnya dalam bidang rekayasa genetika atau genetic engineering, kini memungkinkan untuk dibuat virus sesuai dengan kemauan kita. Apakah dia mau dijadikan vaksin yang sangat bermanfaat untuk kesehatan, atau dijadikan senjata pemusnah massal atau weapon of mass destruction yang sangat mengerikan. Saat ini bioteknologi telah memungkinkan untuk dibuatnya penyakit baru atau new emerging disease, atau penyakit yang direkayasa atau re-emerging disease. Inilah yang disebut dengan dual use, yaitu di satu sisi dapat digunakan untuk bidang kesejahteraan atau peaceful uses, di sisi lain bisa disalahgunakan untuk tujuan lain atau hostile purposes. Bagaimana itu bisa terjadi? Mari kita saksikan video berikut ini.
We may develop technologies that could detect known agents at long range. But in the meantime, science has moved on. There is now the potential to create new and unknown diseases. Weapons that could be used to create a genocide. At the U.S. Army's Biological Research Center, scientists work to develop vaccines to protect against bioweapons. Until now, the range of diseases they were fighting against has been limited. But the new technology of genetic engineering has radically changed the situation.
When you look at current biological weapons, they have many shortcomings. They may be difficult to absorb. They may only survive in perfect weather conditions. So genetic engineering can be used to make that biological agent, that virus or that bacteria, more resistant, more likely to infect people.
Genetic engineering can improve existing agents, and it can also create completely new diseases. Inside a virus or bacteria, its DNA sequences determine how it behaves. It's now possible to introduce new genes into the DNA and change the nature of the organism. By mixing and matching genes, a whole range of designer diseases could be invented. The breakthroughs in genetic techniques in the last 20 years have opened the door to a new era of germ weapons.
The revolution in the biosciences that we've seen since the 70s is every bit as profound as the nuclear revolution of 70 years ago, and could have quite devastating impacts on the future biowarfare and bioterrorism threat.
Relatively simple modifications could transform the way an organism behaves. For example, a disease that currently affects animals could be adapted to be lethal to humans. Over the last 50 years, there has been a successful worldwide program to stamp out smallpox. The final samples of the virus that causes this highly contagious disease are on the point of being destroyed. The greatest concern about the spread of Russian secrets is the claim that they invented a new generation of untreatable diseases. Genetically altered bugs which can overcome immunity through vaccination or resist treatment with antibiotics. Designer bugs are worrying, but they still face the problem of delivery. They could infect the wrong people. But genetic engineering hasn't stopped there. According to the British Medical Association, it may soon be possible to target germs so that they only attack certain people, people with a particular gene or set of genes, while leaving everyone else unharmed.
One hears talk about the development of a kind of ethnic weapon, a weapon that can be targeted to be effective against a particular racial group or group of people.
Research aimed at fighting crime is uncovering new genetic information. At the FBI crime labs in New York, forensic scientists have stumbled across some of the data that could be used to target population groups. Identifying individual people from samples of hair or blood has become possible by looking at human DNA. The National FBI database contains over 100,000 samples. They use them to analyze particular sequences in the DNA called junk DNA. Variations in these sequences now allow the FBI to distinguish one person from another.
The differences that we look for are regions of DNA that may vary in length between people, or they may actually vary in sequence between people also. Sometimes these areas of DNA, because they didn't have a known function, were called junk DNA. But I like to think of the expression, one man's trash is another man's treasure. And for the forensic scientists, these regions, because they vary, are sources of vast information for us to use in forensics.
The forensic work has had an unexpected side effect. The FBI databases uncovered genetic markers which identify African Americans, Hispanic Americans, Caucasians, and Native Americans. Any ethnic group might be identifiable. As well as the FBI work, scientists across the world collaborating in research called the Human Genetic Diversity Project are discovering more and more about our genes and how they make us different. Added together, all the information could be used to make biological weapons targeted at specific people.
Well, if you add together a number of different markers for different populations, you can start to become relatively specific. So, for example, the way in which we handle milk lactose is different between the Asian and European populations. If you then add in blood groups, ABO blood groups, very different distributions. If you also add in height, skin color, and so on, you start to be able to become specific to your target population.
Once genetic markers have been uncovered, a virus could in theory be designed to look for them on entering the body. It would scan the DNA of cells looking for the markers. If it finds them, it inserts itself into the host DNA and starts the process of infection. In this way, the virus would be targeted to attack only people who had the right genetic markers. In Dallas, Texas, a poison is being targeted to attack cancer cells. The doctor in charge of the research is hoping to adapt a toxin that in the past has been used as a biological weapon.
Well, ricin is a natural toxin produced by the castor bean plant, which you can see here behind me. It's an ornamental plant used in many people's homes for decoration, and it has been used for many, many years in espionage. It's been used in all sorts of criminal activities where people wish to eliminate somebody. And the reason it's so effective is because it takes virtually less than you can see in the palm of your hand to kill an individual.
But Dr. Vitetta is hoping to use ricin to save lives.
Ricin could potentially help all cancer patients. It could potentially help patients with a whole variety of other diseases where you know what the target cell for the disease is.
She has managed to modify the ricin poison so that it attacks only the cancerous cells in the body, leaving the healthy ones untouched. It does this by recognizing genetic markers. The ricin is attached to an antibody which looks for receptors on the cell surface. Because of a genetic mutation, cancer cells have different receptors from normal ones. The antibody recognizes them and attaches. Only then is the ricin activated to kill the cell. Successful genetic targeting is one of the golden prizes of medicine today. Millions of dollars are being invested in it in the hope of finding an answer to some of our biggest killer diseases. But it is the very technology that would be needed to identify and kill specific groups of people.
The technology that will develop useful new vaccines, new drugs in a medical sense is going to be very closely associated with the technology which could put that same knowledge to malign use to producing weapons.
An organism that could target specific people would be the coming of age of biological weapons. For the first time, there would be no risk of infecting the wrong people, removing the problem of delivery once and for all. More useful than a nuclear bomb, it would leave land, buildings, and your own troops untouched. Because of these technological advances, the next 10 years could produce a targeted virus or bacteria of this type. Selecting its victims by distinct racial groups, it would be the world's first ethnic weapon. A biological agent that could successfully bring about a genocide.
Genetic targeting is possible, probably within the next 5 to 10 years. The manufacture of relatively specific biological weapons, which are lethal in small volumes, would be possible within that time period, and that we have therefore a very short opportunity, a timescale, to put in place proper control mechanisms to make sure these weapons are never developed.
Even if we can prevent the development of genetic weapons, the spread of existing technology is going to be increasingly difficult to control. Terrorist groups are likely to become more educated in the technology of biological weapons. Dictators and rogue nations will be tempted to level the playing field with the invisible weapons that can kill millions. Since the Second World War, it has been the age of the nuclear bomb. But we are entering a new age, an age where the threat from biological weapons will loom ever larger.